Hello, students. This is Mr. Austin. Today we're going to go through our very first video of the school year, of the first of many, many videos that you will hopefully be watching very thoroughly throughout the school year. But we're going to jump right into it today. We're going to look at our very first unit this year, which is Unit 0, about experimental design and what experiments look like in science and specifically in biology, and how we collected data and what we do with that data and how we analyze that data using statistics. And all of this stuff, I call it Unit 0 because it's going to lay the foundation for this entire school year, because what we are about to look at in Unit 0, today we're just going to go through Topic 1 in this video, but everything in these notes you're going to see intertwined and come up again and again and again in every single unit. So you can think of it as Unit 0, or you can think of it as a foundation to Unit 1, 2, 3, 4, 5, 6, 7. Everything in here you're going to see on tests, future assessments, future assignments, on the AP exam. It's all there. And so this is some really important stuff that we have to start with the school year with. And so let's go ahead and look at Topic 1, which is controlled experiments. So we're going to talk about what a controlled experiment is and the different components of it, and hopefully get you familiar with pinpointing different things from a controlled experiment when you look at an example of one in class, or when we actually do one in a lab or something this year. So it says that a controlled experiment is a scientific test done under controlled conditions. And so what this means is that this is an experiment where we are trying to basically keep everything the same between all the different groups we're testing, and we only want basically just one, ideally just one thing changing during our experiment. And so there's only one variable that we're purposefully making different in the different groups that we're testing in our experiment, but we want everything else to be very controlled, very much the same. So that's what a controlled experiment is. And when you do that, you get some very strong and convincing data from that. And so in a controlled experiment, this is what we're going to focus on in these notes. There's five components that you need to become very familiar with what they are and how to identify those components in an example. And so we're going to look at that right now. So those are the 5 things we're going to talk about. We're going to get into that in a second. Before we do that, just to preface this whole experiment thing, before an experiment, usually there's some kind of observations that are involved of the natural world around us, especially in biology. Scientists are constantly observing specific things, and then those lead to scientific questions. And from those scientific questions, they form a hypothesis, which we'll talk about towards the end of these notes, but that's a testable prediction of what might be the answer to that question. And so in a controlled experiment, we usually develop that experiment and make it look a certain way alongside what our hypothesis is, in order so that experiment is actually going to answer and test whether or not our hypothesis might be correct or not. And then between all of that, there's usually a lot of research that's done in real biological experiments, where you're going to have to research what's already been done in similar experiments in order to come up with a very good experiment and a good educated hypothesis, not just something random that you really put a lot of thought and research into. And so in these notes on Topic 1, we're going to focus on a very specific example of an experiment that I made up. This is not real. I'll go ahead and tell you that there's no fish harmed in this experiment because I never did this experiment, but we're going to use this fake example to kind of try to understand on a basic level what these different things are in an experiment. And so here's the example. Suppose I observe that my pet goldfish behaves strangely when I change his water. And so I start thinking about this and reflecting on it, and I hypothesize that the change in the fish's behavior, it might have something to do with the new water always being colder than the old water. And that change in behavior that I am seeing in my fish when he starts behaving all weird, I think it might be linked to his respiration rate, in terms of how much he's breathing in and using his gills to get oxygen from the water. And so after some research, I find that respiration rate of a fish can be measured actually by counting how many times its operculum which are its gill covers, flap open each minute. And so kind of like your respiration rate is how many breaths you're taking per minute. For a fish, specifically for a goldfish and lots of other fish, they have this structure. It's called a gill cover or an operculum, which covers their gills, but it actually flaps open and close as water is being intaken by the fish and using it to breathe. And so I could count how many times it's opening and closing to figure out its respiration rate and how many breaths it's taking per minute. And so I'm going to go ahead and set up a controlled experiment to test this idea. Let's say I use 5 fish in my experiment who are all the same species, exactly the same type of fish. They're all roughly the same size, basically the exact same size fish. They are all initially placed in a large fish bowl together with water that has a temperature of 20 degrees Celsius. And then what I do is I set up 5 small fish bowls, and each small fish bowl, contains water with a different temperature. One of them has 10 degree water, 15 degree water, 25 degree water, and 30 degree water. And then other than that, the water temperature of all 5 small bowls, other than the water temperature, all 5 small bowls are identical. So it's the same size water, same size bowl, same amount of water in each of those bowls. It's just the temperature that's different. And so I'll show you a little diagram in a second that summarizes what I'm doing. But during the experiment, here's what I do. I'm going to measure the respiration rate of each of those fish in the large bowl at first. I'm going to see how much they're breathing and using their gills in that large bowl with 20 degree water. And then I'm going to immediately measure their new respiration rate once I move them to one of the small bowls, which has a different water temperature. And I'm going to repeat that with the remaining fish in the small bowls. And I'm going to use the data to figure out how much their respiration rate is changing for each of their fish. Are they starting to breathe faster, slower? I'm going to actually measure their respiration rate before and after I change the water temperature and use that to figure out, okay, how much did their respiration rate change? And so this is all summarized right here. So here's my 5 fish right here. Feel free to name them. I kind of, on the previous page, noticed that there was a typo. I didn't say all 5 of these bowls, but one's 10 degrees, one's 15 degrees, one's 20 degrees, one's 25, and one's 30. And so I'm going to measure, they're all starting with the same temperature of water. I'm going to measure the respiration rate. I'm going to put them into one of these new temperatured water bowls, and I'm going to figure out how much their respiration rate changes. Does it go up, down, and by how much? And so here's my results here, my fake results. One of those fish was put in 10 degree water, and its respiration rate actually went down 15 breaths per minute. So it actually started to breathe slower by 15 breaths per minute. Like that was the change in respiration rate. The one that was placed in 15 degree water, its respiration rate also went down. It went down 10 breaths per minute. And then there's a bowl that had 20 degree water, which I forgot to mention on the previous slide. So there's the bowl starting with 20 degrees and then a new bowl, which also has 20 degree water. And when I put a fish in that bowl, its respiration rate actually remains basically the same. It doesn't change. This doesn't mean that it's not breathing. It just means that its respiration rate didn't slow down or go faster. It's changing 0. And then I have another fish who was placed in 25 degree water, and its respiration rate actually went up 15 breaths per minute. And then lastly, one that went into 30 degree water, and its respiration rate went up 25 breaths per minute. So there's my experiment, a summary, a nice little flow chart of what I did in my fake example, and the data from those fish. And so with this experiment, we're going to now talk about the different components of my controlled experiment. And so I don't know if you guys noticed, but after looking at the results for my experiment, I'm going to conclude, I'm thinking that fish that were placed in colder water, it looks like they showed a drop in their respiration rate. Their respiration rate started to slow down. And the fish that were placed in warmer water, they actually, it looks like the respiration rate went up. And you can kind of see that in the data here. And so like I said, let's get into each of those different components now and talk about what those components are in my fake experiment. So let's start with the independent variable. This is, by the way, stuff that should sound super familiar because you're supposed to start learning this at least in middle school science, and then relearning it almost every year of science, but we forget a lot. So, or maybe we don't fully understand sometimes each year, but let's go through. This year is the year you're going to understand what all this stuff means. starting with the independent variable, that is going to be the variable, or sometimes we call it the factor. This is the thing that the experimenter, me in this case, the researcher, is controlling and changing on purpose between the different groups being tested. So think I for independent variable, and I the experimenter, am like purposefully changing it between the groups. So in these different groups that I'm testing, I'm purposefully making this thing different. That's the independent variable. Now, if you think about my goldfish experiment, the thing that I purposefully made different between the 5 groups that I set up, the 5 goldfish that I was testing, was the water temperature and how much the water temperature was changing, if it was getting warmer and how much it was getting warmer, if it was getting colder. The change in water temperature was what I was manipulating, and I purposefully made different between my different groups. That is the independent variable. The dependent variable, this is the variable that I'm predicting is going to change in response to the independent variable. So the independent variable is the water temperature changing. And I'm thinking in this experiment that their respiration rate is going to change because of that. And that's what I'm going to see to find out. And so the dependent variable in my fish example would be how much the respiration rate is changing. Because I'm thinking that the fish's respiration rate is going to depend on how much the water was changing, which I did on purpose. Now, their respiration rate, I don't have any direct control over the respiration rate. I can't point to a fish and say, you start breathing 5 breaths faster. But I'm going to measure this as a result of the experiment that I set up to see how it's changing from my experiment. That's the dependent variable. It's usually, basically always, the thing that you're measuring as a result of what you've set up. In this case, I'm measuring the respiration rate. That's the dependent variable. Now, there's things called constants or controls. They're kind of synonymous words. We also call them control variables. So we had independent variable, dependent variables, and then there's what we call control variables, or we just call them controls or constants for short. Now, these are all the other variables that I purposefully kept identical between all the groups that I was testing. These are all the other things that I made sure were not different between my different groups of fish. And so for my goldfish example, here's a few things. For example, the fish species, they was all the same fish. It's not like I used a goldfish and I tested a beta fish for the next group, and I tested I'm blanking on fish species, another fish, Nemo, on another fish bowl. It was all the same type of fish. They were all the exact same species of goldfish. They were all the same size of fish. They were all in the same original tank that was identical for all of them. They all started off at the same temperature of 20 degrees. And then when I put them in small new tanks, those small new tanks were all the same size and had the same amount of water. The only thing that was different was the water temperature. But all these other things that I just listed, these are what we call control variables. These are things that did not change between all of my fish. These are super, super important. And if you have a really good experiment, you're usually going to have a huge, long list of control variables. And the reason you want that is because if you all these control variables, then when I look at my data and I look at how these fish started breathing differently, I know it's not because of any of these things, because that fish and this fish, it was the same fish species. It's not because they're different fish species that they're breathing differently and responding to the water differently. It's not because they're different size fish. It's not because this fish was put in a bigger bowl and this fish was put in a smaller bowl that was changing the respiration rate. I know it has nothing to do with that. And when I go home and show my mom this cool experiment that I did, she's not going to look at it and be like, well, how do you know that fish isn't breathing faster because you put it in a smaller bowl and it was freaking out, you know? And I'm going to tell my mom, actually, Mom, that was a control variable and they were all put in the same size fish bowl. And so your experiment's going to be stronger and stronger the more control variables you have, because again, in a controlled experiment, the whole point is to only have one thing, ideally one, sometimes 2 or 3, but usually just one thing changing. And so, like I said, the fish's change in respiration rate, when I look at my data, I know, I'm pretty confident that it's not affected by these variables because they were all kept the same for every fish that was being tested or every group that was being tested. And then the last 2 things on here are experimental groups and control groups. An experimental group, that's a group that you've set up that received some version or manipulation of the independent variable, which sounds weird, but it's actually super easy to think about. It's just there can be one or more experimental groups. In mine, I actually had a few experimental groups. Any group that received the independent variable, which in this case was the temperature of the water changing, any group that had a change in water temperature, that's an experimental group. That's a group that I'm physically manipulating the independent variable, the water temperature. I'm changing it on purpose, a colder, warmer. That's an experimental group. So from the fishbowl example, or the goldfish example, the 10-degree water, the 15-degree water, the 25-degree water, and the 30-degree water, those small fish bowls and the fish that were placed in them, those are my experimental groups. So I actually had four experimental groups where I was manipulating the independent variable, the water temperature. Now, a control group, you might already be thinking ahead, that's a group that does not receive the independent variable, or you're not going to manipulate the independent variable for that group. And so this group is going to act as a baseline that lets the experimenter see if the independent variable actually had an effect on the data. And so, and again, just like experimental groups, some experiments will have one control group, sometimes they'll have multiple control groups, and then sometimes, not very often, you might not need a control group, or you can't really set up a control group. But most of the time, there's going to be a control group, one or more control groups. Now, for my goldfish example, I don't know if you noticed, there was one fish that was placed in a fishbowl where the water temperature was the same as it started off with. Like, the water temperature did not change. It went from 20-degree water to 20-degree water. And so in that group, I'm not messing with the independent variable, the water temperature. That is a control group. And I'm going to use that to compare my data to. And it's going to help make my data more meaningful and more convincing to people. If I go back to this outcome here, let's just say I didn't have a control group. So let's pretend this fish doesn't exist and I never tested 20-degree water. So I had some fish that were in colder water and then warmer water. You might be thinking, you're like, well, Mr. Austin, even without this fish, it looks pretty clear that their respiration rate is changing and slowing down when it's colder and speeding up when it's warmer, and you don't really need this. But here's the thing. If I didn't have this fish at 20 degrees, someone could, let's say someone argues with you, your friend, they look at your experiment, they're like, well, how do you actually know? Like, maybe there was something you didn't think of that was causing it to change temperature. And they can't really pinpoint something specific, but let's just say they're questioning you. Or how about we do this? Let's pretend that you did test 20-degree water and the respiration rate did change a lot. So you took a fish from 20-degree water and you put it in a new bowl of water that was also 20 degrees. And let's pretend that the respiration rate actually went up 30 breaths per minute, like a lot. It changed the most. And so along with all this other data, at that point, once you test that, when you're comparing what happened to these other fish to that fish where the water temperature didn't change, now you might start questioning yourself and your experiment. Maybe this is telling you that the water temperature might not be the only thing affecting the fish's respiration rate and how much it's changing, because you have a fish here where the water temperature didn't change and it changed a lot. And if you had not tested that group, you would have thought that, yeah, it was clearly the water temperature having an effect on these fish. But if I set up this group, this control group, and if the respiration rate did change a lot, well, now I might be second guessing myself and starting to think about other things. Maybe the fish are just stressed out because you took them out of the water and put them in a new bowl, or the way that you transferred the fish, or maybe it's just random how the respiration rate is changing. And so now your data is going to start revealing something different when you compare it to those results. But in this case, I did test it and it didn't change really at all, the respiration rate, which is only solidifying my original hypothesis that, yeah, actually, I do think water temperature is affecting the respiration rate. So that's why control groups are very important. They not only make your data more meaningful and more convincing. If you don't test it, sometimes when you do use a control group, you actually find out something that you weren't thinking about, or something that might reveal a different conclusion than you might have originally thought. So those are control groups. And then to make this slightly more complicated, there is 2 different types of control groups that you guys need to know for this class. There's something called negative control groups and positive control groups. Negative, they're both control groups, and they're both used to create a baseline of comparison. Okay, they are control groups, and you're going to use these groups, these special groups, to compare what's happening to your other groups to reveal important things. Now, in a negative control group, I'll start off by saying that's usually the more common one, and so you're going to see that more often than positive control groups. A negative control group is actually like the one I just described. This is a control group that's not, it's not going to receive anything additional that the other fish did not get. Okay, so it's exactly the same as all the other groups. This group that you tested, it's exactly the same as all the other groups. The only thing different is that they were not given the independent variable. That's why we call it a negative control group, because it's all the same except for this one thing was taken away from them. Like they did not get a change in water temperature, but everything else was exactly the same. The way you transferred the fish bowls, all that stuff, the type of fish, the same. And they didn't get anything extra. Okay, like it's just the same. They're lacking this one thing. Now in a positive control group, these get a little interesting sometimes. These do receive some kind of extra treatment that the other groups did not get. So this is a group, a control group, that actually gets something extra that no one else got. And that extra treatment that it's going to get is usually something that is going to produce some kind of expected, predetermined outcome for that group. This will make sense in a second, hopefully. But these, they are identical to the experimental groups, except there's 2 things that make them different than the other groups. First of all, they usually don't receive the independent variable, which means in this group, they're not going to receive any manipulation of the independent variable, just like a negative control group. So the independent variable is not going to, we're not going to apply it to this group. So if for the fish example, like their water temperature would stay the same. We're not going to change the water temperature. But then there's going to be something else different. The other thing that's going to be different about a positive control group is that it's going to receive some other treatment or variable that is different than all the other groups. And the reason we do that sometimes is it allows us to make sure that the experimental setup and the way that we're doing the experiment and the equipment that we're using as part of our experiment and the way that we're collecting our data and the method of collecting our data, that all of that is working the way we think it's working, and it's working in a way that nothing is going wrong that we're not aware of. Like this is just to double check that everything's working correctly in your experiment. So here's an example of a negative and positive control group. A negative control group, that's like the one I just explained in the goldfish example, the one that was put in 20-degree water, that came from 20-degree water, so the water temperature didn't change and didn't receive anything extra. That's a negative control group. Now, a positive control group, which we didn't have one actually, we didn't have a positive control group in our experiment because you don't always need to have each of these. It just depends on the experiment. But let's say if I did want a positive control group, here's maybe what it might look like. I could set up a 6th fish and small bowl. Let's say I added to my experiment where the water was also 20 degrees in that small bowl. So just like the negative control group, it's also 20-degree water where the water temperature is not going to change. But in this bowl, I also give it a little bit of caffeine I dissolve in the water. And so that's something that none of the other groups got, none of the other fish got, is the caffeine. And I'm adding caffeine to the water because I specifically know after doing my research that it's already been predetermined and known that if you dissolve a little bit of caffeine in the water, the fish respiration rate will increase. It causes the respiration rate to go up. And the reason I'm going to do this in my experiment is because I want to make sure, just by default, that things are happening the way they're supposed to happen. So if everything's working in my experiment correctly and there's nothing weird affecting these fish, then if I give this one fish caffeine, its respiration rate should go up because it's already been determined by scientists and other experiments that like you give fish caffeine, their respiration rate goes up. And so that allows me to make sure that there's nothing preventing the fish's respiration rate from changing. And so if I do test that, let's say I did test a positive control group, and I find when I add caffeine, it actually doesn't cause the fish's respiration rate to go up. That would then be a warning to me that something might be affecting my data that I'm not aware of. And so maybe the method of me transferring the fish and stressing them out is affecting how their respiration rate is changing, or them confined to this bowl. Maybe it's too small of a bowl and it's really hindering their respiration rates changing in a natural way because of water temperature. And so it helps me figure out, or maybe the way I'm measuring their respiration rates, like by me visually counting how many times their fish flaps are opening, that I'm actually doing that accurately. If I don't have this positive control group, I won't know for sure if everything I did and set up wasn't hugely affecting my data or incorrectly causing me to get bad data. So that's what a positive control group is. And then the very last thing here, and I know this is running on 25 minutes here, so stop right after this, is what makes a good hypothesis. And so in this class, you guys will be writing hypotheses throughout the school year for different experiments that we do. And so there's some components of what a good hypothesis that makes it a lot better than some pretty terrible hypotheses. And so a good hypothesis, again, this is a statement in which you're predicting a specific outcome. Here are some components or characteristics of a good hypothesis. It needs to be testable. It needs to use the independent and dependent variables, and it needs to make a specific prediction about the outcome of an experiment. And so you might have been taught this if-then method, like in middle school especially, they like to do that for students. It helps them a lot, where if you use an if-then statement, it actually does help you make a better hypothesis that meets these expectations. If you're not familiar with an if-then statement, it's basically when you write for your hypothesis, you write, if this is done in my experiment and I set up these fish or whatever, then this is going to happen, right? Like you're specifically saying what you're about to do and what you think is going to happen. Now you don't have to, in reality, you don't have to use an if-then statement. It's not always, sometimes it makes it harder to write the hypothesis if you're forcing yourself to do an if-then statement, but it's definitely okay to do one. But let me give you two examples of a hypothesis. One of them is a not so good one, and one of them is a better one that meets these requirements. So here's an example of a weak hypothesis. If the water temperature changes, then the respiration rate of the fish will change. Now that is testable. Just by reading that, I can think of how I could set up an experiment to test that, kind of like the one I just did. It does use the independent variable and the dependent variable that I think I would be using for my experiment, where like I'm going to be changing the water temperature and I'm going to be looking at their respiration rate. But the problem with this hypothesis is it's not making a specific prediction. It's a very generic, general prediction. It's saying if you change the water temperature somehow, in some way, then the respiration rate of the fish will change somehow, in some way. So it technically is a prediction. It's just a very not specific prediction. And so here's an example of a stronger one. It says, if the water temperature decreases, then the respiration rate of the fish will increase. That is very specific. So again, that's testable. I can think of a way to test that pretty easily with an experiment that is using the independent and dependent variables that I think I'm going to use for my experiment, talking about temperature of the water changing and the respiration rate of the fish. And this one is specific. You're specifically making a claim, like you're being gutsy and you're saying like, I think specifically if the water temperature gets colder, the fish's respiration rate will go up. Or maybe you think it's going to be the opposite or something else. But at least you're being specific. That is a great hypothesis. This one right here, great. Okay, good, very good. A+ to this hypothesis. Now, were they correct? Who cares? We'll find out in the experiment. Just because you're wrong doesn't mean it was a bad hypothesis. But when you look at the experiment, it does look like the data does not support their hypothesis. It's actually the opposite, it looks like, from the data. But that's fine. That's something that you will conclude then, that your hypothesis was actually not supported and actually the opposite was supported. That's great. That's how science works. But that's still way better than just generically saying something, you know? So anyway, you guys have questions to answer in these notes on the left side of your notes. So go through that. There is some questions that are going to ask you to click on this link on the slides. So on the slides, if you pull them up on my website, you're going to click this link, and it's a short example of a real experiment, an easy one that you're going to answer some questions about towards the end of these questions in topic 1. And so anyway, good luck with that. If you have questions, you can definitely ask me next time you see me in class. But that's it for our first video. See you guys later.